What is hidden in the Great Barrier Reef? 13 Secrets and Wonders. Think the Great Barrier Reef is simply the planet's most stunning underwater postcard? A place for colorful fish, clear water, and perfect vacation photos? Throw every tourist brochure and basic geography lesson you ever read straight out the window. This isn't just a pretty diving destination. It is the single largest living structure ever built on Earth. And what's really happening inside it will leave even the most experienced marine scientists speechless. Today, I'm sharing 13 revelations straight from the latest field research and satellite observations. Stick with me all the way to the final secret, because you're going to discover exactly how this colossal living organism quietly produces a huge portion of the oxygen we breathe every day, and why rising ocean temperatures could quietly rewrite the chemistry of our atmosphere. Ready to go beneath the surface? Kicking off our list is the single most mind-bending fact most people never stop to consider. Have you ever looked at satellite photos of the planet and wondered what that long, pale ribbon snaking along the northeast coast of Australia actually is? From 230 miles up, it looks like a delicate turquoise thread stitched across dark blue ocean. Zoom in closer and the thread becomes thousands of individual reefs stitched together. Closer still, and you realize this is no ordinary landscape. It is alive, growing, breathing. The Great Barrier Reef stretches more than 1,400 miles from the northern tip of Queensland almost to Papua New Guinea. That is roughly the driving distance from New York City to Miami and back again. It covers an area roughly the size of Italy or the state of New Mexico. And every square inch of that enormous footprint was constructed, particle by particle, by tiny animals smaller than your little fingernail. These animals are hard corals, more than 400 different kinds working together like master masons over thousands of years. Each coral polyp builds a limestone skeleton around itself, then clones itself again and again. Generation after generation, the skeletons stack up until entire mountain ranges rise from the sea floor. Recent surveys by NASA and the Australian Institute of Marine Science confirm this is the only structure created by living organisms that is clearly visible from low Earth orbit. No forest, no coral atoll, no kelp forest comes close. The reef has been growing in roughly the same location for at least 500,000 years, surviving multiple ice ages when sea levels dropped so low that parts of it became dry land. Every time the ice melted and the ocean rose again, the coral simply grew upward, keeping their sunlit tops just below the waves. Picture standing on a dive boat at dawn. The water is mirror calm. You slip over the side and descend slowly. At first, the reef looks like rolling green hills covered in velvet. Then sunlight hits, and suddenly every surface explodes into color. Electric purples, neon greens, deep crimsons, soft lavenders. It feels like swimming through an underwater flower garden that never stops blooming. Gentle currents carry the faint scent of salt and something sweeter, almost like fresh-cut grass. You hear the soft crackle of snapping shrimp, the distant thump of parrotfish biting coral, the low whoosh of water moving through channels. Everything is in motion, yet everything feels perfectly balanced. To understand the true scale, consider this. If every person on Earth stood shoulder to shoulder, we still would not cover the surface area of the Great Barrier Reef. Researchers estimate it supports more than 9,000 species of fish, 6 of the world's 7 marine turtle species, more than 30 species of whales and dolphins, and at least 1,400 different kinds of coral-building organisms. That biodiversity is not random. It exists because the reef creates thousands of different microhabitats, shallow lagoons protected by outer ramparts, deep channels where strong currents bring nutrients, sandy bottoms where garden eels sway like living stalks of wheat. Each zone supports its own specialized community, and together they form one of the richest ecosystems our planet has ever produced. But the most astonishing part is how slowly and deliberately this living architecture grows. On average, the framework of the reef thickens by about 1 to 2 millimeters per year in the fastest growing areas. Over centuries, that adds up to several feet. Over millennia, it becomes mountains. Think of it like the slowest, most patient construction project in history. No cranes, no blueprints, only sunlight, warm water, and the quiet determination of countless microscopic architects. And right now, in this very moment, that construction is still happening. New polyps are still settling, still secreting calcium carbonate, still reaching toward the light. Why does any of this matter beyond the postcard beauty? Because this single living structure plays roles far beyond its own borders. It acts as a massive carbon sink, locking away billions of tons of carbon dioxide in its limestone skeleton. It shelters juvenile fish that later stock commercial fisheries across the entire western Pacific. It buffers shorelines against storm surges, protecting coastal communities that would otherwise face far worse erosion and flooding. And perhaps most humbling of all, it reminds us that nature is capable of engineering on a scale we can barely comprehend, using nothing more than sunlight and chemistry. We've only scratched the turquoise surface so far. We surfaced from that orbital view and slipped beneath the waves, but the reef still has countless layers left to reveal. Next, we're going to witness the biggest synchronized event on the planet, one that turns the entire ocean into a living snowstorm under the light of a full moon. Claiming second spot is a revelation that turns the whole idea of a quiet underwater world completely upside down. Have you ever stood in the middle of a packed city street at rush hour, watching thousands of people hurrying in every direction, each with their own purpose, yet somehow the whole system flows without total chaos? Now imagine that same frantic, perfectly organized energy, but instead of concrete sidewalks and honking taxis, it's happening under 20 to 30 feet of crystal clear tropical water, with no traffic lights, no bosses, no schedules, just instinct, sunlight, and survival. That is exactly what you step into when you descend onto a healthy section of the Great Barrier Reef. It is not a serene, empty garden. It is one of the busiest, most densely populated cities on the entire planet, and the residents number in the billions. Recent surveys from the Australian Institute of Marine Science and other leading research groups put the total number of different species calling the reef home at around 9,000. That count includes more than 1,600 kinds of fish alone, everything from tiny darting damselfish the size of your thumbnail to massive potato cod that can weigh as much as a grown person. Add in over 600 species of coral, 30 different kinds of whales and dolphins that pass through or live nearby, 6 of the world's 7 marine turtle species nesting on its islands and cays, plus thousands more mollusks, sponges, crustaceans, sea stars, sea cucumbers, worms, and sea snakes. To put that in perspective, coral reefs cover less than 1% of the ocean floor worldwide, yet they support roughly 25% of all known marine life. The Great Barrier Reef alone punches way above its weight class in that statistic. Picture gliding slowly along the edge of a bommy, one of those massive coral heads that rise like underwater mesas from the sandy bottom. Sunlight pours through the surface in bright shafts, turning the water into liquid gold. Suddenly, a school of several hundred fusiliers flashes by in perfect formation, their silver-blue bodies catching the light like polished chrome. A moment later, a green sea turtle cruises past, ancient and unhurried, munching on seagrass as casually as someone eating chips on a park bench. Parrotfish the color of tropical punch scrape algae off the coral with loud, crunching bites you can actually hear through your mask. They are literally eating the reef and pooping out fine white sand that helps build the beaches. A clownfish pokes its head out of an anemone's tentacles, daring you to come closer, while cleaner wrasses hover like tiny doctors waiting for their next patient. The soundscape is just as alive. There is a constant low crackle from snapping shrimp hiding in crevices, popping their specialized claws to stun prey with tiny sonic booms. Parrotfish grind coral with beak-like mouths, producing that distinctive crunch. Triggerfish grunt warnings when you get too close. At night, the reef quiets down a little, but then you hear the soft whoosh of water moving through channels and the occasional distant boom of a bumphead parrotfish headbutting a coral head to break it open for food. Every sense gets flooded: the cool silk of water against your skin, the faint salty taste on your lips, the bright chemical smell of healthy reef mixed with a hint of something floral from spawning algae. This incredible density did not happen by accident. The reef creates an astonishing variety of living spaces in a relatively small area. Outer reefs face the open ocean with crashing waves that bring fresh nutrients and oxygen. Inner lagoons are sheltered, calm nurseries, where juvenile fish hide among branching corals. Sandy channels act like highways for larger predators. Vertical walls drop into deeper water where currents concentrate plankton like a buffet line. Each microhabitat supports specialists: butterfly fish that nibble only certain polyps, gobies that live symbiotically with pistol shrimp, moray eels that hunt at night in the shadows. The whole system is a masterclass in niche partitioning. No two species compete for exactly the same resources at exactly the same time. And the genetic library this place represents is staggering. Scientists estimate that more than 30% of the world's coral reef fish species can be found here. A single location holding such a huge slice of global marine diversity. That makes the reef not just a local wonder, but a planetary treasure chest of evolutionary adaptations. Lose pieces of it, and you lose irreplaceable blueprints for how life solves problems in warm, shallow seas. Beyond the sheer wow factor, this living metropolis delivers real benefits to people far beyond its edges. Tourism alone brings in billions of dollars every year to Queensland's economy and supports tens of thousands of full-time jobs: dive guides, boat captains, hotel staff, restaurant workers, airplane pilots flying visitors in from around the world. Healthy reefs mean healthy fish stocks that feed commercial and subsistence fisheries across the Western Pacific. The natural breakwater formed by the outer reefs protects coastal towns from storm surges and erosion, saving potentially billions more in infrastructure damage during cyclones. Yet the most profound value might be the quiet lesson in cooperation. Every fish, every polyp, every shrimp plays a role. Remove too many herbivores and algae overgrow the corals. Lose the cleaners and parasites spread. Disrupt the nutrient cycles and the whole balance tips. It is a reminder that complex systems thrive on connection, not isolation. A principle that applies just as much to human societies as it does to underwater cities. We have just wandered through the bustling streets of this underwater metropolis, but the reef never sleeps. It throws the biggest party of the year once the conditions are exactly right, and when it does, the ocean turns into something out of a dream. Next, we are going to witness billions of creatures synchronizing their most intimate moment to the phases of the moon. Rounding out the top 3 is perhaps the most magical, synchronized spectacle nature puts on anywhere on Earth. Imagine slipping into the ocean on a warm tropical night, just a few days after the full moon has lit up the sky like a giant silver coin. The water is ink black except for the faint glow filtering down from above. You descend slowly, your dive light piercing the darkness like a narrow beam through fog. At first, everything seems calm. The usual gentle sway of soft corals, the occasional flash of a fisheye reflecting your light. Then, without warning, the reef comes alive in the most extraordinary way. Tiny pinkish bundles start rising from every surface. They look like miniature floating marshmallows or upside-down jellyfish, drifting upward in slow motion. More and more appear, thousands, then millions, until the water column turns into a gentle blizzard of glowing pink snow. The bundles burst open near the surface, releasing clouds of eggs and sperm that swirl together in milky slicks stretching across the ocean for miles. The scent is faint but unmistakable, a mix of fresh ozone and something almost sweet, like overripe fruit carried on the current. You hear nothing dramatic, just the soft hiss of water moving and the distant pop of bundles breaking apart. But visually, it is overwhelming. The entire sea seems to be giving birth to itself under the cover of night. This is the annual mass coral spawning on the Great Barrier Reef, often called one of the planet's greatest natural wonders. Researchers from James Cook University first documented the full scale of this event in the early 1980s, though local divers and traditional owners had observed pieces of it for generations. What makes it so remarkable is the precision timing. Hundreds of coral species, more than 400 different kinds, release their gametes within the same narrow window of a few nights each year. Inshore reefs often go first in late October, while outer reefs peak in November or sometimes early December. The trigger is a perfect alignment of environmental cues. Water temperatures must stay above roughly 27 degrees Celsius for weeks to mature the bundles. The full moon provides the gravitational and light signal, and calm conditions after sunset minimize dispersal before fertilization can happen. Each healthy colony can release millions of egg-sperm bundles, pink, orange, or white packets, bundled together like tiny balloons. A single large table coral might produce hundreds of millions in one night. Across the entire reef system, estimates run into the trillions of gametes released during peak spawning nights. The bundles float upward because they contain lipid droplets that act like natural buoyancy aids. Once at the surface or mid-water, they rupture, freeing the eggs and sperm to mix in the plankton-rich layer. Fertilization happens externally. Sperm swim frantically to find compatible eggs from the same species. Successful unions form tiny planula larvae that drift with currents for days to weeks before settling on suitable substrate to start building new skeletons. Why synchronize like this? Evolution has fine-tuned the strategy for maximum success in a risky environment. If corals release gametes randomly throughout the year, most would be wasted, eaten by plankton feeders, or diluted too much to find partners. By spawning en masse, the odds skyrocket. The water becomes saturated with reproductive material, turning fertilization rates from near zero to surprisingly high. Different species often stagger their exact nights by a day or two to reduce crossbreeding, ensuring genetic purity within kinds. It is nature's version of the world's largest, most perfectly timed speed dating event, happening simultaneously across more than 1,400 miles of reef. Scientists monitor these nights obsessively. Teams from the Australian Institute of Marine Science and other institutions dive after dark with collection nets, gathering spawn slicks for laboratory study. They track which colonies participate, measure bundle quality, and assess overall reef health. A strong spawning event, with dense slicks and high fertilization, signals resilience. Even after recent stress periods, many sections still produce impressive displays, showing the system retains reproductive power. In recent years, these collections have fueled cutting-edge restoration work. Researchers fertilize eggs in controlled tanks, rear larvae, and deploy them back onto damaged areas using innovative techniques like larval seed boxes that boost settlement rates dramatically. Picture the aftermath at dawn. The surface shimmers with iridescent slicks that catch the first light. Seabirds wheel overhead, feeding on floating debris. Fish swarm the upper layers, gorging on nutrient-rich gametes. The event feeds the entire food web, a brief but massive pulse of energy that ripples outward for months. Plankton blooms follow, supporting larval fish, which in turn feed bigger predators. It is a reminder that reproduction here is not just about making more corals. It sustains the whole interconnected web of life. This synchronized explosion of life stands as proof of the reef's enduring ingenuity. In a world where many ecosystems struggle to recover from disturbance, the ability to orchestrate such a vast, coordinated renewal every year is extraordinary. It gives hope that, with protection and smart intervention, the Great Barrier Reef can keep regenerating itself. We have just witnessed the reef's greatest annual celebration, billions of tiny lives launched into the currents under moonlight. But as the spawn drifts away, the water deepens and darkens. Next, we plunge into hidden voids beneath the surface, places where entire forgotten worlds have waited for millennia in near total darkness. Dropping to fourth is where things get genuinely mysterious. Places on the reef that feel like they belong to another world entirely. We have celebrated the surface colors, the bustling crowds, and the moonlit spawn clouds. Now imagine turning away from the sunlit shallows and descending into sudden, perfect darkness. The water turns from turquoise to deep indigo, then to inky black within just a few dozen feet. Your dive light becomes the only source of truth. Ahead, the reef floor simply vanishes. A wide, circular hole opens beneath you, edges sharp and vertical, walls dropping straight down like the inside of a giant stone well carved by time itself. These are the blue holes of the Great Barrier Reef, submerged sinkholes that plunge far deeper than the surrounding seafloor, hiding ecosystems that have evolved in near isolation for thousands of years. The largest and best studied of these features reach depths of 200 to 300 feet or more in places where the main reef platform sits only 60 to 80 feet deep. Formed during periods of much lower sea levels tens of thousands of years ago, when vast areas of the continental shelf were exposed to air and rain, acidic groundwater dissolved the limestone bedrock in classic karst fashion, the same process that created famous cave systems on land. When the ice ages ended and the oceans rose again, these sinkholes flooded, becoming vertical oases trapped below the main reef. Some connect to networks of tunnels and caverns that stretch for miles underground, while others are steep, isolated pits sealed off except for the narrow opening at the top. Picture the descent. You fin slowly downward along one wall, light beams sweeping across jagged limestone etched with ancient scallops and solution tubes. The water feels noticeably cooler here, several degrees lower than the sun-warmed surface layers, and the current is almost non-existent, creating an eerie stillness. Visibility can drop from crystal clear to 20 or 30 feet as fine silt stirred by your fins drifts like slow smoke. At around 100 feet, the light from above fades to a dim twilight blue. Deeper still, and it becomes absolute night, except for whatever your own lights reveal. The walls are alive in unexpected ways. Clusters of deep-water corals that glow faintly under ultraviolet. Tube sponges stretching like organ pipes. Delicate fans of black coral swaying in the faintest upwelling currents. Schools of small fish, squirrelfish with huge eyes adapted to low light, hover motionless, then dart away in unison when your beam sweeps over them. What makes these blue holes truly extraordinary is how dramatically different their inner worlds are from the surrounding reef. The upper rims blend seamlessly with typical coral gardens, brain corals, staghorns, plate corals, all thriving in full sunlight. But drop below the lip, and conditions change fast. Light intensity plummets. Oxygen levels can dip in the deeper layers during stratification periods, and nutrient concentrations spike from organic matter sinking in from above. In some holes, researchers have measured near anoxic pockets near the bottom, zones where oxygen is so scarce that only specialized microbes and a handful of hardy invertebrates survive. These anaerobic communities include bacteria that metabolize sulfur compounds, producing faint hydrogen sulfide scents detectable by sensitive instruments. In one documented case from a northern section of the reef, a blue hole contained dense mats of microbial life, forming thick, slimy biofilms across the floor. Ecosystems more akin to deep-sea vents than to a tropical coral reef. Recent expeditions using remotely operated vehicles and technical divers have mapped dozens of these features in detail. Teams from James Cook University, the Australian Institute of Marine Science, and international partners have cataloged unique species found nowhere else on the reef. Certain sponges, sea squirts, and small crustaceans appear restricted to these dark refuges. Some holes harbor relict populations of fish that retreated there during past environmental shifts and never left. The isolation acts like a natural laboratory. Evolutionary pressures inside differ so sharply from the sunlit exterior that speciation has occurred over millennia. One study published in a leading marine biology journal describes several new species of polychaete worms and amphipods collected exclusively from within a single blue hole, their closest relatives living hundreds of miles away in completely different habitats. Beyond pure discovery, these hidden pockets hold practical promise. The extreme conditions, low oxygen, high nutrients, unusual chemistry, force organisms to develop novel biochemical defenses and enzymes. Compounds isolated from deep-hole sponges and bacteria have already shown activity against certain bacteria and cancer cell lines in preliminary lab tests. While no miracle drugs have emerged yet, the potential is real. Similar extreme environments on land and in the deep sea have yielded antibiotics, anti-inflammatories, and painkillers in the past. Every new blue hole explored adds another page to that chemical library. Yet the very isolation that makes these places so fascinating also makes them fragile. They act as natural sediment traps, accumulating fine particles and organic debris over centuries. Any major disturbance, heavy anchoring nearby, increased runoff carrying fertilizers, or changes in regional water quality can smother the delicate communities inside. Technical diving to these depths carries real risks: nitrogen narcosis, decompression sickness, equipment failure in total darkness. Exploration is careful, deliberate, and always guided by strict conservation protocols. Stand at the rim of one of these voids on a bright afternoon and look down. The hole looks like a perfect circle of midnight set into turquoise. Schools of baitfish swirl above the drop-off like silver confetti, while a lone eagle ray glides along the edge before banking sharply and vanishing into shadow. It is a stark visual reminder that even in one of the most studied marine ecosystems on Earth, entire hidden realms remain almost untouched, waiting for the next careful visitor with lights and curiosity. We have peered into these sunken cathedrals, felt the chill of their depths, and glimpsed lives shaped by darkness. But the reef has been building its story far longer than any of us have been watching. Next, we travel back through layers of time itself to see how this living wonder has outlasted ice ages, sea level swings, and entire human civilizations. Fifth in line takes us far beyond the present moment, back through layers of deep time to see how this living wonder has quietly outlasted entire chapters of Earth's history. Walk with me for a second through a thought experiment. Picture yourself holding a core sample pulled straight from the heart of the reef, a long cylindrical tube of drilled limestone about the thickness of your forearm. In your hands, you are literally holding pages from a book that has been writing itself for 500,000 years. Each centimeter of that core represents decades, sometimes centuries, of slow accretion. The colors shift subtly from pale cream to warmer beige to darker bands where sediment or organic matter settled during rougher times. Under a microscope, those bands reveal microscopic skeletons stacked one atop another, like the rings inside an ancient tree trunk. Only these rings are made of calcium carbonate, laid down by countless generations of coral polyp. Geologists and paleoclimatologists have extracted and analyzed hundreds of such cores from across the Great Barrier Reef over the past several decades. The longest and most carefully studied reach back more than 500,000 years, well into the Pleistocene epoch when our own species had not yet appeared. During that span, the planet cycled through at least 5 major ice ages. Global sea levels dropped by as much as 400 feet during the coldest peaks, exposing huge areas of what is now the continental shelf. Parts of the modern reef platform became dry land, karst hills, river valleys, even patches of forest and grassland. Yet every time the climate warmed, ice melted, and the oceans rose again, the coral simply resumed growing upward from whatever high points remained submerged. They tracked the rising water surface layer by layer, keeping their sunlit living tissue just below the waves where photosynthesis could continue. This ability to migrate vertically with sea level change is one of the reef's most impressive survival tricks. In places where the underlying substrate was stable, the upward growth reached staggering thicknesses, more than 100 feet in some cores. Where the foundation shifted or subsided, the corals adjusted sideways or found new footing. The result is a geological record so detailed it rivals ice cores from Greenland or sediment layers from ocean basins. Scientists can read past sea surface temperatures from the oxygen isotope ratios locked inside the coral skeletons. They can track periods of higher river runoff from spikes in terrestrial sediment. They can even detect ancient bleaching events preserved as thin stress bands, where growth slowed or symbionts were temporarily expelled. One particularly revealing core sequence shows the reef enduring the last glacial maximum around 20,000 years ago. At that time, sea level was roughly 400 feet lower than today. Much of the current reef was high and dry. Yet as warming began around 18,000 years ago, the oceans rose at rates sometimes exceeding 3 feet per century. The corals responded by accelerating vertical accretion in favorable spots, reaching growth rates of up to half an inch per year during the fastest pulses of inundation. By about 7,000 years ago, the modern reef framework had largely re-established itself across the flooded platform. That means the structures we dive on today are, in many places, relatively young compared to the full history. Mere teenagers built atop the bones of much older ancestors. This long view changes how we think about resilience. The Great Barrier Reef is not a fragile newcomer. It is a veteran that has weathered multiple global-scale climate shifts, far more extreme than anything recorded in human history. It survived rapid warming episodes that raised temperatures several degrees Celsius above modern averages. It endured sea level oscillations that would drown or strand entire coastlines today. Through all of it, the basic blueprint: symbiotic algae powering coral animal hosts, massive colony sizes, efficient calcium carbonate secretion, remains stable enough to keep rebuilding. Of course, past success does not guarantee future survival. The conditions the reef faces now differ in important ways. Previous warm periods unfolded over thousands of years, giving time for gradual adaptation and range shifts. Today's ocean warming is compressed into mere decades, driven by human greenhouse gas emissions. Ocean acidification was not a factor during past interglacials because carbon dioxide levels never climbed as high or as fast. Nutrient pollution from agriculture and coastal development adds stresses that ancient corals never encountered. Still, the deep time record offers genuine grounds for cautious optimism. The fundamental biological machinery has proven extraordinarily durable when given sufficient time and space. Researchers continue to mine these ancient archives for lessons applicable right now. By comparing growth rates, stress bands, and geochemical signatures across millennia, they build models that forecast how different sections of the reef might respond to projected warming scenarios. Cores also serve as natural benchmarks, showing what healthy accretion looked like before large-scale human influence. In restoration projects, understanding which coral types dominated during past recovery phases helps guide which species or genotypes to prioritize when replanting damaged areas. Hold that core sample again in your mind. Feel its weight. See the faint color changes that mark wet seasons, dry spells, cyclones, quiet decades. Every millimeter tells a story of persistence against odds that make our current challenges, while serious, seem part of a much longer conversation between life and a changing planet. The Great Barrier Reef has been keeping score for 500,000 years. We are only just beginning to read the book properly. We have traveled back through 500,000 years of quiet endurance, reading the reef's own diary written in stone. But even in the present day, the reef still finds ways to surprise us with beauty we can barely explain. Next, we turn on special lights and watch the entire structure light up like it is hosting an underwater rave. Colors and patterns that exist for reasons far beyond human eyes. Sixth revelation flips the script on what we think we see during the day. Because when the right lights hit, the entire reef transforms into something straight out of a science fiction dream sequence. Picture this. You are on a night dive, but not with ordinary white beams. Instead, your team switches to powerful ultraviolet or blue light torches, the kind marine biologists use to reveal secrets invisible in normal daylight. The moment those lights sweep across the coral heads, the world changes. What looked like muted browns, greens, and pinks under sunlight suddenly erupts in electric hues. Vivid lime green branching arms pulsing like neon signs. Deep purple plates glowing with an otherworldly shimmer. Bright cyan tentacles waving as if lit from within. Pops of hot pink and electric yellow scattered across the landscape. It feels like someone flipped a switch and turned the reef into an underwater nightclub. Silent, slow motion, and impossibly vibrant. This phenomenon is biofluorescence, and it is far more widespread and sophisticated than most people realize. Corals produce special proteins related to the famous green fluorescent protein family, the same molecules that earned a Nobel Prize in chemistry for their discovery in jellyfish back in the 1960s. In reef-building corals, these proteins absorb high-energy blue or ultraviolet light from the sun and re-emit it at longer, lower-energy wavelengths we perceive as green, yellow, cyan, red, or orange. Recent work by researchers studying corals on the Great Barrier Reef has documented dozens of distinct fluorescence patterns across species, some uniform across the whole colony, others concentrated in tentacles, oral discs, growth edges, or even specific polyps within a single head. In certain massive corals like Porites, for example, fluorescence can appear as starry spots, broad blankets, or delicate rim highlights, varying not just between species, but between individual colonies and even different strains living side by side. Why go to all this biochemical trouble? The leading explanations center on protection and optimization in a high-light environment. Shallow tropical waters are flooded with intense sunlight, including harmful ultraviolet rays that can damage delicate tissues and the symbiotic algae living inside them. Fluorescent proteins act like a built-in sunscreen. They capture potentially damaging short-wavelength light and convert it to safer, longer wavelengths that scatter harmlessly or even get used by the algae for photosynthesis. Studies have shown that corals in brighter, more exposed positions often express higher levels of these proteins, ramping up production when ultraviolet exposure increases. In deeper or shaded spots, the glow is subtler, but still present, suggesting a flexible toolkit that helps corals fine-tune light across their tiny bodies. Another intriguing role involves luring in the symbiotic algae themselves. Tiny dinoflagellates called symbiodiniaceae need to find and settle into coral hosts to survive. Experiments have demonstrated that green fluorescence emitted by the coral host can attract these microscopic partners, acting like a welcoming beacon in the plankton soup. Once inside, the algae photosynthesize and share sugars with the coral in return for protection and nutrients. The classic win-win partnership that powers most of the reef's growth and color. Fluorescence may help seal that deal early on, especially for young corals recruiting new symbionts after settlement. And then there is the visual signaling side. Some researchers propose the glow helps with camouflage or communication in low-light conditions, perhaps confusing predators at dawn or dusk, or even attracting certain prey like zooplankton that mistake the light for food sources. In deeper habitats, where blue light penetrates best, fluorescence turns otherwise drab surfaces into glowing lures, drawing in small organisms that corals can capture with their tentacles. It is not hard to imagine the evolutionary advantage. A coral that advertises just enough to snag extra meals, without exposing itself to too much risk, gains a competitive edge. The sheer variety is staggering. Surveys across the Great Barrier Reef have cataloged fluorescence in hundreds of species, with emission peaks spanning the visible spectrum. Some corals glow steadily, others pulse or change intensity with water movement or light cycles. Under controlled lab conditions, scientists have isolated and sequenced genes coding for these proteins, revealing families with subtle tweaks that shift colors by just a few nanometers, enough to create the dazzling patchwork we see on night dives. This molecular diversity mirrors the reef's overall biodiversity. One more layer of adaptation that helps explain why so many coral kinds can coexist in such close quarters. Beyond pure wonder, these glowing molecules have real-world applications far from the ocean. Fluorescent proteins from marine organisms revolutionized biomedical imaging decades ago, allowing researchers to tag specific genes, proteins, or cells in living tissues and watch processes unfold in real time. Coral-derived versions continue to inspire new tools, brighter, more stable variants for tracking cancer cells, monitoring plant stress, or even developing sensors that detect environmental pollutants. Every time a diver marvels at a neon green acropora branch under blacklight, they're witnessing chemistry that has already changed how medicine sees inside the human body. Yet the glow is fragile in its own way. Stress from warming waters, pollution, or acidification can disrupt protein production or cause uneven expression, sometimes leading to those eerie fluorescent patches that appear during early bleaching stages as a last-ditch protective response. Healthy, thriving colonies tend to show balanced, vibrant fluorescence. Stressed ones can look patchy or overly intense in certain colors. Night surveys using ultraviolet lights have become a non-invasive way for researchers to assess colony health quickly. A glowing reef is often a resilient one. So next time you see photos of a daytime reef looking beautiful but somewhat subdued, remember what happens after dark with the right illumination. The Great Barrier Reef is not just colorful, it is literally engineered to glow, turning invisible energy into visible magic. For reasons that protect, attract, and sustain life across scales we are only beginning to understand. We have just switched on the blacklights and watched the reef throw its private light show, a hidden layer of beauty and biochemistry at work every single day. But all that color and energy depends on an invisible partnership that is more delicate than it seems. Next, we zoom in close to meet the tiny roommates living inside every coral polyp, the microscopic algae whose sudden departure can turn paradise pale and silent. Seventh Secret gets right to the fragile heart of the whole show. Because none of the color, the glow, the spawning clouds, or the massive living structure would exist without an invisible partnership that is as essential as it is delicate. Zoom in as close as science allows. Past the waving tentacles, through the translucent tissue of a single coral polyp no bigger than a pinhead. Inside its body, packed into special cells like guests in a luxury hotel, live millions of microscopic algae. These tiny plants, each about the width of a red blood cell, are members of a group called zooxanthellae, though modern researchers prefer the more precise name Symbiodiniaceae to reflect the diversity of strains now recognized. They are not random hitchhikers. They are full partners in a deal that has powered reef-building corals for tens of millions of years. Here is how the arrangement works in everyday terms. The coral animal cannot photosynthesize on its own. It has no chlorophyll, no way to turn sunlight directly into food. Instead, it provides the algae with a safe home, protected housing inside its own tissues, plus a steady supply of carbon dioxide from respiration, inorganic nutrients recycled from waste, and a prime, sunlit location high on the reef where light is abundant. In exchange, the algae use sunlight to photosynthesize, producing sugars and other organic compounds. They share up to 90% of that freshly made food with their coral host, enough to cover most of the animal's energy needs for growth, reproduction, and building the limestone skeleton that forms the reef framework. It is one of the most efficient solar-powered systems in nature. Sunlight hits the coral surface, gets captured by the algae inside, gets converted to usable energy, and keeps the entire colony alive and expanding. Picture it under a microscope. The polyp's transparent body glows faintly with the golden-brown hue of densely packed algae cells. When light streams in, you can almost see the tiny chloroplasts inside the algae working, churning out oxygen as a byproduct, which the coral uses for its own respiration. The oxygen bubbles sometimes form visible streams rising from healthy coral heads on calm days, a subtle sign that the partnership is humming along perfectly. The coral even controls where the algae live, concentrating them in the uppermost tissue layers to maximize light capture, shading them slightly during peak sun to prevent damage from too much intensity. This symbiosis is not static. Different coral species host different genetic varieties of algae, and a single colony can shuffle its population of symbionts over time in response to changing conditions. Some strains perform better in bright, warm water. Others tolerate cooler temperatures or lower light. This flexibility acts like an internal insurance policy. If one type struggles, others may take over, helping the coral weather short-term stress. Researchers have watched this shuffling happen in real time during mild warming experiments. Colonies that successfully adjusted their algal communities showed far less tissue loss than those locked into less adaptable partnerships. But here is where the fragility creeps in. The bond is voluntary on both sides. There is no physical tether holding the algae inside. Under normal conditions, the coral expels a small percentage of its symbionts daily as part of routine housekeeping, then welcomes fresh ones from the water column. When the coral experiences stress, especially prolonged high water temperatures, it triggers a protective response. It expels the algae en masse. Without their partners, the coral tissues turn ghostly white because the golden brown algae gave the color in the first place. This is coral bleaching. The animal can survive for weeks or months on stored fats and whatever zooplankton it can catch. But without photosynthesis, it slowly starves. If temperatures drop back to normal quickly enough, surviving algae or new recruits from the water can repopulate the tissues and color returns. Recovery can take years though, and repeated or severe events leave scars. Slower growth, higher disease susceptibility, reduced reproduction. Field studies across the Great Barrier Reef have tracked these dynamics for decades. In healthy years, bleaching is rare and localized, a few colonies here and there, usually in the hottest shallows. But during marine heat waves, the scale becomes staggering. The widespread bleaching events of recent years showed how quickly the partnership can break down when ocean temperatures stay elevated for too long. Sections that lost their algae early in a heat spell sometimes recovered partially by shifting to more heat-tolerant strains, demonstrating that the symbiosis retains some adaptive wiggle room. Yet the more frequently these disruptions happen, the harder it becomes for colonies to bounce back fully before the next stress arrives. Understanding this relationship has driven some of the most promising conservation work on the reef. Scientists are now mapping which algal strains confer the greatest thermal tolerance, then testing ways to introduce those hardier partners to vulnerable corals before heat waves hit. In lab and field trials, corals seeded with heat-resistant symbionts have shown significantly better survival during simulated warming. Other approaches focus on strengthening the coral side of the partnership, selecting genotypes that maintain tighter control over their algae, or expel them less readily under moderate stress. Every breakthrough here is built on the same core insight: protect the symbiosis, protect the reef. So when you look at a thriving coral head pulsing with life, vivid, growing, feeding the fish around it, remember the invisible workforce inside. Those billions upon billions of microscopic algae are the quiet engine room. They turn sunlight into food, oxygen into breath for the animal host, growth into the limestone bones of the world's largest living structure. Disrupt that engine, and the lights start to dim. We have just met the tiny roommates that keep the entire reef powered and colorful, a partnership so intimate that losing it turns vibrant life pale and still. But even this ancient alliance has vulnerabilities that outside forces are starting to exploit. Next, we meet an unexpected predator that can sweep across the reef like a slow-moving storm, devouring living coral at rates that leave entire sections stripped bare. Eighth on the countdown brings us to a creature that looks almost comical at first glance, but when its numbers explode, it turns into one of the reef's most destructive natural forces. Imagine gliding over a once vibrant patch of branching coral in the middle of the day. The water is warm, sunlight pours through in bright shafts, and everything should be alive with color and motion. Instead, the scene feels wrong. The branches that used to wave soft purple and green are now stark white skeletons, stripped clean, no tissue left, no algae glowing inside. Scattered across this ghostly landscape are dozens upon dozens of large, spiny sea stars, each one the size of a dinner plate or bigger. Their bodies are a mottled mix of red, purple, and orange, covered in long, venomous spines that glint in the light. They move slowly, methodically, arms draped over living coral like slow-motion octopuses. Where their tube feet touch healthy tissue, you can watch the coral polyps retract in panic, then dissolve as digestive enzymes pour out from the sea star's stomach. It is not a fast attack. It is patient, relentless, and eerily quiet. The only sounds are the faint crunch of spines against skeleton and the soft hiss of water moving past. These are crown of thorns sea stars, named for the ring of sharp, toxin-tipped spines circling their central disc like a medieval weapon. Scientifically known as Acanthostare plantae, they are native to Indo-Pacific reefs, including every major section of the Great Barrier Reef. In low numbers, they play a useful role. Natural pruners that feed mainly on fast-growing, plate-like corals, preventing any one species from dominating space and allowing slower-growing branching types to compete. A single adult might eat a few square feet of coral tissue per year under normal conditions, keeping the ecosystem in gentle balance. The problem starts when their populations surge into what researchers call outbreaks. During these outbreaks, the density can jump from a handful per hectare to hundreds or even thousands in the same area. One documented wave in the 1960s and 70s removed up to 90% of living coral cover from some northern and central reef sections over just a few years. More recent surges, tracked meticulously since the 1980s, have shown similar patterns. Populations explode, feed voraciously, leave white skeletons behind, then crash when food runs out or predators increase. A healthy adult crown of thorns can consume up to 10 square feet of coral tissue annually, sometimes more during peak feeding. Multiply that by thousands of individuals moving together in loose aggregations, and entire reef zones can be stripped in months to a couple of years. What sets off these booms? The triggers are now much better understood thanks to long-term monitoring programs run by the Australian Institute of Marine Science and partner universities. Nutrient runoff from coastal agriculture and urban development plays a starring role. Excess nitrogen and phosphorus wash into the sea during wet season floods, fueling massive plankton blooms. Crown of thorns larvae, tiny free-swimming planktonic forms, feed on that plankton soup. Higher food availability means more larvae survive to settle on the reef and grow into adults. Warmer waters speed up larval development and increase survival rates too. Once established, fewer natural predators, giant triton snails, some triggerfish, and a handful of larger fish can keep numbers in check when densities stay low. But outbreaks quickly overwhelm those controls. The visual impact is heartbreaking. Healthy coral gardens turn into marine graveyards. White branches still standing but lifeless. Soft tissues gone. Skeletons slowly colonized by turf algae that give the surface a fuzzy, brownish-green haze. Fish that relied on the complex structure for shelter and food start to disappear or move elsewhere. Herbivores struggle as the algae they graze shifts from nutritious macroalgae to less palatable turf. The whole food web feels the ripple. In severe cases, sections that once supported dense schools of colorful reef fish become quiet. Open expanses where only a few hardy species remain. Control efforts have evolved significantly. Since the 1980s, dedicated crown of thorns control programs have removed hundreds of thousands of sea stars, using trained divers with collection bags and, more recently, bile salts injected directly into the animal. A method that kills quickly without harming other marine life when used carefully. These culls target high-density areas during early outbreak stages, buying time for corals to recover before the next generation of larvae arrives. In some regions, the intervention has kept coral cover 20 to 30% higher than it would have been without action. Community involvement has grown too. Citizen science programs train recreational divers to spot and report outbreaks, feeding real-time data into management maps. Still, crown of thorns outbreaks are not isolated events. They interact with other stresses. A reef already weakened by bleaching recovers much more slowly when sea stars move in to feed on the remaining live tissue. Combined pressures create feedback loops, where one disturbance makes the next more damaging. Long-term data show that while outbreaks have occurred naturally for centuries, ancient reef cores contain layers of crown-of-thorns spines. The frequency and severity appear to have increased in recent decades, tied to human-driven changes in coastal water quality and ocean temperatures. Watching a crown of thorns feed up close is strangely mesmerizing and deeply unsettling at the same time. The sea star averts its stomach through its mouth, spreading it like a thin sheet over the coral surface. Digestive juices pour out, breaking down living tissue into a soupy meal that the animal absorbs. The coral has no defense beyond retracting polyps and producing mucus, tactics that buy minutes or hours, not days. It is nature's slow, methodical cleanup crew gone rogue, reminding us that even native species can become overwhelming when the balance tips. We have just witnessed one of the reef's own residents turn predator on a massive scale. A natural process amplified by human influence into something far more destructive. But the threats keep building. Next, we face the silent thief that steals color from the water itself, a phenomenon that has left vast stretches of the reef looking like bleached bones under the sun. Ninth wake-up call hits harder than anything we have covered so far, because this thief works in total silence, stealing color from the water itself and leaving behind landscapes that look like they belong to a different planet. Picture flying over the Great Barrier Reef on a clear morning in late summer. From above, the water sparkles turquoise and electric blue, broken by the darker outlines of individual reefs and the white foam of breaking waves on the outer edge. Everything looks alive and perfect, the postcard version everyone expects. Now imagine the same flight a few months later during one of the hottest summers on record. Patches that used to glow with vibrant coral hues have turned ghostly pale. From the air, those sections appear almost bone white against the surrounding blue, as if someone drained the life out of them overnight. Drop lower, get closer, and the change becomes heartbreaking. Entire fields of branching staghorn corals, massive brain corals, plate corals, all standing exactly as they did before, but stripped of every trace of color. The polyps have retracted or died back, the golden brown algae that gave them life are gone, and what remains are pure white calcium carbonate skeletons, bleaching under the relentless sun. This is coral bleaching, not a disease, not an attack by predators, but a stress response so severe it can push entire reef systems to the brink. When ocean temperatures climb and stay elevated for weeks, often just 1 or 2 degrees Celsius above the long-term summer maximum, the symbiosis we explored earlier breaks down. The coral expels its zooxanthellae algae in a desperate bid to survive what it interprets as toxic conditions. Without those algae providing up to 90% of its energy through photosynthesis, the coral animal enters survival mode. It draws on stored fats, catches whatever plankton it can with its tentacles, but the clock starts ticking. If the heat lingers too long, tissues starve, polyps die off, and the skeleton is left exposed. The white you see is not new bone. It is the bare framework that was always there, hidden beneath living tissue in the algae that colored it. Recent marine heat waves have made this process tragically visible on a massive scale. During the consecutive bleaching events starting in 2016, large parts of the northern and central Great Barrier Reef lost significant live cover. Aerial surveys flown by dedicated monitoring teams documented the extent in stunning, sobering detail. Mile after mile of once vibrant reef turned pale. Follow-up dives confirmed that roughly 29% of the total coral cover across the entire system disappeared between 2016 and the early 2020s, with bleaching as the primary driver in many areas. Some reefs in the far north saw losses exceeding 50% in a single season. The scale is hard to overstate. These are not isolated patches. They are vast underwater landscapes, shifting from living ecosystems to skeletal ruins in a matter of months. Recovery is possible, but painfully slow. In places where water temperatures return to normal relatively quickly, and few other stresses piled on, some colonies regained color within a year or two, as surviving algae or new recruits from the water column repopulated the tissues. Heat-tolerant strains sometimes took hold, giving those corals a slight edge against the next warming pulse. Yet full structural recovery, regrowing complex three-dimensional habitats that support fish, invertebrates, and the entire food web, takes a decade or longer even under ideal conditions. Branching corals that provide the most shelter grow slowly. Massive boulder corals can take centuries to reach full size. When bleaching hits repeatedly, as it did in quick succession during recent years, many colonies never get the breathing room to rebuild. The system tips towards simpler, flatter landscapes dominated by weedy algae and fewer habitat specialists. What makes the situation urgent is how these events are no longer rare anomalies. Ocean warming driven by greenhouse gases has raised baseline sea temperatures across the tropics. What used to be an extreme summer now occurs more frequently. Satellite data from agencies tracking global sea surface temperatures show that the number of days above bleaching thresholds has increased sharply in the past 4 decades. For the Great Barrier Reef specifically, consecutive marine heat waves have compressed recovery windows to the point where some scientists describe parts of the system as approaching ecological ratchets. Once certain thresholds are crossed, shifting back becomes exponentially harder. The human fingerprints are clear. While natural climate variability has always caused occasional warm spells, the rapid intensification we see today aligns with measured increases in atmospheric carbon dioxide and the resulting ocean heat uptake. Nutrient pollution and overfishing can worsen outcomes. Stressed corals bleach at lower temperature thresholds, and fewer herbivores mean faster overgrowth by algae on damaged skeletons. Yet the dominant signal remains temperature. Reducing local pressures helps. Cleaner water, protected herbivore populations, targeted restoration. But the only path to long-term stability runs through global emissions reductions that limit warming. Seeing a bleached reef up close changes your perspective forever. The silence is the worst part. No crackle of snapping shrimp, no crunch of parrotfish, no darting schools of fish. Just white branches swaying gently in the current, already starting to be colonized by thin films of turf algae. The water feels emptier, the light harsher without the soft diffusion of living tissue. It is not dramatic explosion, it is quiet erasure. Yet even here, hope flickers. Restoration teams are replanting nursery-grown corals selected for resilience. Scientists track recovery in real time with drones, divers, and autonomous underwater vehicles. Every surviving colony that regains color is data point proving the system still has fight left. We have watched the color flee and the reef turn pale. Under heat it was never built to endure for so long. The silence of those white skeletons echoes louder than any storm. But the forces at work do not stop at temperature alone. They reach deeper, into the very chemistry of the water itself. Next we face the slow acid rain falling invisibly across the entire ocean, dissolving the reef's foundation grain by grain and turning growth into a losing battle. 10th Alert pushes us into territory that feels almost unfair, because this threat does not roar in like a storm or crawl across the bottom like a predator. It arrives invisibly, molecule by molecule, changing the very chemistry of the water until building and maintaining a skeleton becomes a losing battle for the corals themselves. Imagine standing on the edge of a healthy reef flat at high tide. The water laps gently against branching corals that stretch toward the surface like delicate candelabras. Polyps extend their tentacles to feed. Algae inside their tissues glow golden brown in the sunlight, and the whole colony feels solid, alive, expanding year by year. Now fast forward a few decades into the future under business-as-usual carbon emissions. The same coral heads still stand in roughly the same places, but something fundamental has shifted. The branches look thinner, more brittle. New growth at the tips is slower, patchier. When you run a finger along a living surface, carefully of course, the skeleton feels softer than it should, almost chalky. Break off a small piece underwater and it crumbles more easily than healthy coral ever would. This is not bleaching. This is dissolution, the slow chemical erosion of the very limestone framework that defines the reef. The culprit is ocean acidification, driven by the ocean absorbing roughly 1/4 to 1/3 of all carbon dioxide humans release into the atmosphere each year. When carbon dioxide dissolves in seawater, it reacts to form carbonic acid, which lowers the pH, making the water more acidic. Since the start of the industrial era, the average surface ocean pH has dropped by about 0.1 units on the logarithmic scale. That may sound small, but because pH is logarithmic, each unit represents a 10-fold change in acidity. So 0.1 means the ocean is already about 30% more acidic than it was 200 years ago. Projections from comprehensive climate assessments show that by the middle of this century, under moderate to high emissions pathways, acidity could rise another 50 to 100% relative to pre-industrial levels. Why does acidity matter so much to corals? Reef-building corals construct their skeletons from a mineral called aragonite, a form of calcium carbonate that is particularly sensitive to changes in seawater chemistry. To build those skeletons, corals must maintain a state called supersaturation. The water around them needs to hold more calcium and carbonate ions than are needed to keep aragonite stable. When the ocean becomes more acidic, carbonate ion concentration drops sharply. The water moves closer to, or even below, the saturation point where aragonite can form, or even remain intact. Below a certain threshold, known as the aragonite saturation state, new skeleton growth slows dramatically. In extreme cases, existing skeletons begin to dissolve faster than the coral can repair or add to them. Recent field measurements and controlled experiments on the Great Barrier Reef tell a clear story. In areas where aragonite saturation is still comfortably above 3, the level many researchers consider healthy for robust calcification, corals grow at expected rates. But in zones already dipping toward or below that line during certain seasons, calcification rates have measurably declined. Some massive Porites corals, the slow-growing giants that form the backbone of many reefs, show reduced linear extension and density in their annual growth bands over the past few decades. Branching Acropora species, the fast growers that create the most complex three-dimensional habitat, appear especially vulnerable. Their thin, delicate skeletons struggle first when carbonate becomes scarce. The math is stark. Laboratory studies simulating future conditions have shown that at an aragonite saturation state around 2.5, calcification can drop by 20 to 40% compared to today. Push toward 1.5 or lower, plausible by the end of the century under higher emission scenarios, and net calcification turns negative for many species. That means the reef as a whole could start losing skeletal material faster than it builds it. The framework that has taken thousands of years to accumulate would begin to crumble under its own weight and the constant mechanical action of waves, fish, and boring organisms. Structures that once sheltered juvenile fish and buffered coastlines would flatten, simplifying the habitat and reducing biodiversity step by step. This is not a hypothetical far future risk. Early signs are already appearing in the most sensitive locations. Shallow, high-temperature lagoons and reef crests, where natural diurnal swings in pH already push conditions close to the edge. Combined with warming, the double stress is brutal. Heat triggers bleaching that expels algae and halts energy supply, while acidification makes skeleton repair and new growth far more energetically expensive, even when the coral can photosynthesize. The result is a compounding disadvantage that tips the balance from net builder to net eroder. Efforts to understand and counteract this are intensifying. Researchers maintain long-term monitoring sites where they measure seawater chemistry continuously, tracking pH, total alkalinity, and carbonate parameters in real time. These data feed into sophisticated models that forecast how different sections of the reef might fare under various global emissions pathways. On the intervention side, small-scale experiments test whether locally buffering seawater, for example by adding carefully controlled amounts of alkaline substances near nursery corals, can create temporary refuges for vulnerable larvae and juveniles. While these techniques are not scalable across 1,400 miles of reef, they provide proof of concept for protecting high-value restoration sites during critical growth phases. The deeper lesson here is scale and timescale. Carbon dioxide molecules released from a power plant in one hemisphere today can influence seawater chemistry on the other side of the planet decades from now. The ocean has absorbed so much already that even if emissions stopped tomorrow, acidity would continue to rise for years before slowly reversing over centuries. That lag time makes aggressive global emissions cuts not just desirable, but essential if we want future generations to still see a calcifying, growing Great Barrier Reef, rather than a dissolving relic. Stand on that same reef flat in your mind's eye once more. Feel the gentle current. Watch the sunlight dance across living branches. Now realize that beneath every polyp, every tentacle, a quiet chemical war is being waged. One where the weapons are invisible gases and shifting ion balances. Win that war on a planetary level, and the reef keeps building. Lose it, and the skeletons we marvel at today become tomorrow's crumbling ruins. We have felt the slow acid burn eating away at the reef's very bones. A threat so subtle it hides in plain sight until the framework starts to fail. But as these pressures mount, the entire system edges closer to points where recovery may no longer be possible. Next, we confront the accelerating heat that could push large parts of the reef past critical thresholds, turning a resilient veteran into a system struggling to hold on. 11th Horizon marks the point where wonder gives way to something far more serious. The accelerating reality that heat is no longer arriving in isolated waves, but building into a relentless pressure that could push large parts of the reef past recovery thresholds forever. Imagine the reef as an extraordinarily tough, battle-tested organism that has survived ice ages, sea level swings, cyclones, starfish plagues, and everything else the planet could throw at it for 500,000 years. Now picture that same organism facing a stressor that arrives faster, hotter, and more frequently than anything in its deep time record. That is where we stand today with marine heat waves. These are not gentle summer warm-ups. They are extended periods, sometimes months long, when sea surface temperatures climb 1, 2, or even 3 degrees Celsius above the long-term average maximum for that location and season. For corals already living near their upper thermal limits, that extra heat acts like a fever the body cannot shake. The mechanism is brutally straightforward. When water temperatures stay elevated, the symbiotic algae inside coral tissues start producing harmful, reactive oxygen species as a byproduct of photosynthesis under stress. Those compounds damage both the algae and the coral host. To protect itself, the coral expels the algae, the bleaching process we have already seen. If the heat dissipates quickly, many colonies can recover by repopulating with new or surviving symbionts. But when the fever lingers week after week, energy reserves deplete, tissues thin, disease susceptibility spikes, and mortality climbs. What starts as paling turns to full white, then to tissue sloughing, skeleton exposure, and in the worst cases, complete colony death. Recent years have delivered back-to-back demonstrations of how quickly this can escalate. Comprehensive aerial and in-water surveys documented unprecedented consecutive marine heat waves across the Great Barrier Reef. In some northern and central sectors, the cumulative heat stress, measured in degree heating weeks, a metric that combines intensity and duration, reached levels previously considered extreme outliers. Large areas bleached multiple times in short succession, with insufficient time between events for meaningful recovery. The result? Significant net loss of live coral cover in regions that had previously shown strong rebound potential. Certain fast-growing branching species, which provide the most complex habitat structure, suffered the heaviest mortality because their thinner tissues have less energy storage and higher metabolic demands. What makes the situation especially concerning is the compression of recovery windows. In the past, major bleaching years were separated by decades, giving reefs time to regrow framework, recruit new larvae, and rebuild biodiversity. Now, projections from sophisticated climate ocean models show that under current emissions trajectories, severe heat stress could occur every few years by mid-century, and become near annual in some areas by the end of the century. At that frequency, even the most resilient corals struggle to keep pace. A colony that barely survives one bleaching event may enter the next already weakened. Lower reproductive output, reduced skeletal density, higher vulnerability to secondary stressors like crown of thorns feeding or disease outbreaks. The system starts losing its redundancy. Fewer survivors mean fewer larvae in the water column, fewer genetic variants to draw from, slower structural regrowth. Tipping points loom in these models. Researchers have identified critical thresholds, often framed around global average warming levels, beyond which large-scale functional collapse becomes probable. At roughly 1.5 degrees Celsius of long-term global warming above pre-industrial baselines, many projections indicate that 50% or more of current coral cover across the Great Barrier Reef could face annual or near-annual bleaching, with recovery increasingly unlikely in the absence of heroic local interventions. Push toward 2 degrees Celsius global warming, still within range under some emissions pathways, and the outlook shifts dramatically. Functional extinction of reef building capacity in large sections, meaning the system no longer accretes net framework, no longer supports the full suite of dependent species, no longer provides the coastal protection and biodiversity services it has delivered for millennia. These are not alarmist guesses pulled from thin air. They rest on peer-reviewed ensembles of global climate models downscaled to regional ocean conditions, validated against observed bleaching patterns over the past decade. Satellite records from agencies monitoring sea surface temperatures confirm the upward trend in heat wave frequency and intensity. In-water monitoring sites scattered across the reef track coral condition year after year, providing ground truth data that aligns closely with model forecasts. Yet the picture is not uniformly bleak. Pockets of hope persist even under pressure. Some reefs, often in slightly cooler, better flushed locations or those with naturally heat-tolerant algal communities, have shown partial resistance or faster recovery. Selective pressures are already shifting populations toward more thermally robust genotypes and symbiont strains in places that have experienced repeated mild stress. Active interventions are scaling up. Coral nurseries cultivate heat-resistant fragments. Larval restoration deploys millions of lab-reared settlers to damaged areas, and hybrid approaches combine natural recruitment enhancement with targeted shading or cooling during peak heat events. These efforts cannot replace the need for global emissions reductions, but they demonstrate that local action can buy precious time and preserve genetic diversity while the world works toward stabilizing the climates. The existential weight of this moment is hard to overstate. The Great Barrier Reef is more than a collection of pretty corals. It is a planetary-scale ecosystem engineer that has shaped biodiversity, fisheries, coastal protection, and cultural heritage for countless generations. Losing its functional capacity would ripple outward, diminished fish stocks affecting millions of livelihoods across the Indo-Pacific, weakened natural barriers exposing shorelines to greater storm damage, reduced tourism value impacting regional economies, and the irreversible loss of evolutionary lineages that have no equivalent elsewhere on Earth. We stand at a fork. One path continues the current trajectory, incremental emissions cuts that still allow warming to climb past critical thresholds, heat waves arriving too frequently for recovery, and a gradual but accelerating flattening of the reef into simpler, less productive states. The other path accelerates deep, rapid decarbonization globally while doubling down on local resilience measures, giving the reef the best possible chance to adapt, persist, and perhaps even regain ground in a cooler future ocean. Look out over that turquoise expanse one more time in your mind. Feel the warmth of the water, the gentle push of the current, the quiet pulse of life beneath the surface. Then remember, this ancient, resilient system is telling us, through every bleached branch and every surviving colony, that time is no longer on our side. unless we act decisively on the forces driving the heat. We have stared into the accelerating fever that could tip the reef past points of no easy return, a threshold where resilience gives way to irreversible decline. But the stakes stretch far beyond the reef itself. Next, we examine how its potential loss would reverberate through economies, coastlines, and millions of human lives that depend on this living shield. 12th Impact zooms out from the underwater world to show exactly how tightly this living reef is woven into human lives. Far beyond postcards and dive trips, it quietly powers economies, shields coastlines, feeds communities, and supports industries worth billions every single year. Picture the bustling harbor in Cairns or Port Douglas on a bright Queensland morning. Boats loaded with divers, snorkelers, and sightseers line the docks, engines idling as crews check tanks, brief passengers, and hand out wetsuits. Further south, in towns like Airlie Beach or Mackay, the same scene repeats on a smaller scale. These are not just tourist operations. They represent one of the most visible economic engines tied directly to the Great Barrier Reef. Independent economic analyses estimate that reef-related tourism and recreation generate around $6 billion Australian dollars annually for the regional and national economy. That figure covers everything from luxury island resorts and day-trip catamarans to family-run dive shops, underwater photography guides, helicopter flights over the outer reefs, and the hotels, restaurants, bars, and souvenir stores that feed off visitor spending. More than 70,000 full-time equivalent jobs trace back to this activity. Not seasonal gigs, but year-round livelihoods for boat captains, marine biologists leading educational tours, hotel staff, chefs serving reef-inspired seafood dishes, airplane pilots shuttling visitors from Brisbane or Sydney, and marketing teams promoting the destination worldwide. In some coastal communities, the reef accounts for a dominant share of local employment. Lose significant visitor numbers, and entire towns feel the ripple immediately, through reduced retail sales, lower property values, and strained public services. The draw is obvious. People fly from every continent to experience something found almost nowhere else: the chance to swim through living coral gardens teeming with clownfish, sea turtles, manta rays, and sharks, in water so clear it feels like flying. That unique draw supports not just direct reef tourism, but a broader hospitality ecosystem. Airport transfers, car rentals, domestic flights within Australia, and even international cruise ships that include reef stops all benefit. When surveys ask international visitors why they chose Queensland, the Great Barrier Reef consistently ranks as the #1 reason. A single natural wonder acting as the anchor for an entire regional tourism industry. Beyond leisure, the reef delivers protection that saves far more money than it costs to maintain. Its outer ramparts and thousands of individual reefs form the world's largest natural breakwater system. During cyclones, which hit the Queensland coast with regularity, those structures absorb and dissipate wave energy before it reaches the mainland. Engineering studies have quantified the value. The reef reduces wave heights by up to 97% in some sheltered areas, cutting erosion and storm surge damage that would otherwise require massive artificial seawalls, beach renourishment projects, and repeated repairs to roads, homes, and infrastructure. One comprehensive valuation placed the annual coastal protection service provided by the reef at several $billion. A quiet insurance policy paid for by nature rather than premiums. Fisheries feel the benefit too. The complex three-dimensional habitat created by healthy coral supports juvenile stages of countless commercially and recreationally important species: coral trout, red emperor, Spanish mackerel, prawns, crabs, and more. Many adult fish migrate out to deeper waters or adjacent shelf areas where they enter commercial catches. While the reef itself is not the primary fishing ground for large-scale trawlers, its nursery function underpins stocks harvested across broader regions of the Coral Sea and Western Pacific. Healthy reefs mean healthier fish populations over time. Degraded or flattened ones produce fewer juveniles and contribute to declining catches in connected areas. Indigenous communities have known and relied on these connections for tens of thousands of years. Traditional owners across more than 70 Aboriginal and Torres Strait Islander groups maintain deep cultural, spiritual, and practical ties to the reef. It provides food through sustainable harvesting practices passed down through generations, materials for tools and ceremonies, and a living classroom for teaching younger members about seasonal cycles, navigation, and ecological balance. The cultural value cannot be fully captured in dollars, yet it forms an irreplaceable part of identity, well-being, and heritage for those communities. Put all these services together: tourism revenue, job creation, storm protection, fishery support, cultural significance, and independent economic assessments have valued the Great Barrier Reef's annual contribution to Australia at tens of billions of dollars, when indirect and non-market benefits are included. That makes it one of the most economically significant ecosystems on the planet, rivaling major industries in scale while requiring no human-built infrastructure to deliver its output. The flip side is sobering. When coral cover declines, when bleaching leaves large areas pale and slow to recover, when structural complexity flattens, the economic returns shrink in lockstep. Tourism operators report fewer bookings after major bleaching years as images of white reefs circulate globally. Dive sites lose their wow factor when fish schools thin and colors fade. Coastal properties face higher insurance premiums or outright risk when natural wave barriers weaken. Fisheries see longer-term pressure as recruitment drops. Restoration projects, growing corals in nurseries, deploying them on damaged sites, removing crown of thorns, help mitigate losses, but they come with real costs in labor, science, and funding. Every percentage point of live cover preserved or regained translates directly into preserved or regained economic and social benefits. This tight coupling between reef health and human well-being creates powerful incentives for action. Governments, businesses, scientists, traditional owners, and everyday citizens have invested heavily in monitoring, research, water quality improvements, pest control, and climate advocacy because the stakes are measurable in jobs, dollars, and community stability. The reef is not a distant luxury. It is infrastructure, employer, protector, and cultural heart all in one. Think about the next time you see a photo of turquoise water and colorful coral. Behind that beauty lies a quiet economic engine that employs tens of thousands, shields coastlines from storms, supports fisheries, and anchors cultural identities. Damage that engine and the consequences cascade far beyond the underwater world, into boardrooms, bank accounts, and family dinner tables across Queensland and beyond. We have just traced how deeply the Great Barrier Reef is embedded in human prosperity and security, a living asset whose decline would cost billions and disrupt millions. But the full scale of what is at stake reaches even further, into the global climate system itself. Next, we confront the largest, most sobering secret of all: how destabilizing the reef's hidden carbon stores could amplify warming far beyond what most people realize, locking in changes that affect every coastline on Earth. Crowning our list is the single most sobering secret buried deep within the reef, one that turns this living wonder from a regional treasure into a planetary linchpin, whose destabilization could quietly accelerate the very warming that threatens it. Imagine standing on the deck of a research vessel anchored over a quiet section of the outer reef at dusk. The water below looks calm, almost ordinary, gentle swells rolling across coral heads that have stood for thousands of years. But beneath that serene surface, in the thick layers of sediment trapped between the reef framework and the underlying limestone platform, something massive and largely invisible has been accumulating for millennia. Trapped within those fine-grained deposits are vast stores of organic carbon, the remains of dead algae, coral tissue, fish bones, shellfish shells, microbial mats, and plant debris washed in from nearby mangroves and seagrass meadows. This is not just a thin dusting. In places, the sediment column reaches tens of meters thick, and across the entire Great Barrier Reef system, the total buried carbon reservoir is estimated in the range of billions of tons of carbon dioxide equivalent. Recent sediment core studies and geophysical surveys have begun to map the scale more precisely. Researchers drilling into reef interiors and analyzing acoustic profiles have found that these carbon-rich layers act like natural archives, recording past productivity, storm events, and even ancient human influences through isotopic signatures and microfossils. Much of that carbon sits in relatively stable forms under anaerobic conditions deep in the sediment pile, locked away from the atmosphere for thousands to tens of thousands of years. In a balanced system, it remains safely buried, contributing to long-term carbon sequestration and helping regulate global cycles. The danger emerges when warming reaches the seafloor itself. As ocean temperatures rise, slowly but steadily penetrating deeper layers, microbial communities in the sediment respond. Heat accelerates the breakdown of organic matter by sulfate-reducing and methanogenic bacteria. In oxygen-poor pockets, this process releases methane, a greenhouse gas roughly 25 to 80 times more potent than carbon dioxide over 20-year and century timescales, respectively. Even small increases in methane flux from warming sediments could create a feedback loop. More heat drives more decomposition, more methane escapes into the water column and eventually the atmosphere, amplifying global warming and pushing ocean temperatures higher still. Field measurements are already detecting early signals. In several studied locations across the Great Barrier Reef and similar carbonate platforms worldwide, scientists have recorded elevated methane concentrations in near-bottom waters during warmer months. Bubble streams, small but visible releases of gas from the seafloor, have been documented in shallow lagoons and near river-influenced zones where organic input is naturally higher. While current fluxes remain modest compared to major sources like wetlands or thawing permafrost, the concern centers on potential amplification. If large areas of buried carbon become destabilized as bottom temperatures climb just a few degrees, the cumulative release could rival or exceed some terrestrial sources over decades to centuries. This is not a sudden catastrophe waiting to explode. It is a slow burn amplifier, insidious because it operates below the threshold of immediate visibility. Satellite and shipboard monitoring catch surface warming and bleaching long before sediment processes show up in the data. Yet modeling studies that couple ocean circulation, sediment biogeochemistry, and climate projections suggest that under sustained warming trajectories, these stores could transition from net sink to net source within this century in vulnerable carbonate systems. The Great Barrier Reef, with its enormous area and fixed sediment accumulations, sits squarely in that risk zone. The planetary stakes become clear when you zoom out. The reef's buried carbon is part of a broader class of coastal blue carbon reservoirs, mangroves, seagrass meadows, salt marshes, and carbonate sediments that collectively hold more carbon per unit area than many terrestrial forests. Protecting and restoring these systems ranks among the most cost-effective natural climate solutions available. When they remain intact, they sequester carbon long-term and buffer against sea level rise and storms. When destabilized, they can flip to become additional sources of greenhouse gases, making global mitigation targets harder to reach. This feedback potential adds urgency to every layer of protection already in place. Reducing local stressors, improving water quality to support healthier sediment-trapping seagrass and mangroves, controlling nutrient runoff that fuels excess organic deposition, maintaining herbivore populations that prevent algal overgrowth, helps keep the system resilient. Active blue carbon strategies such as sediment stabilization and targeted restoration of adjacent coastal habitats can enhance burial rates and reduce vulnerability. But none of these local measures can fully offset the primary driver: global ocean warming rooted in atmospheric carbon accumulation. Think about the quiet accumulation happening right now, unseen beneath the waves. Every blade of seagrass that dies and settles, every coral fragment that crumbles and gets buried, every algal bloom that sinks. All of it adds to a reservoir that has helped keep our climate stable for millennia. That same reservoir, if destabilized, could quietly tip the scales toward faster warming, higher sea levels, more intense storms, and greater pressure on every coastline, including the very shores the reef has protected for so long. The Great Barrier Reef is not just a beautiful ecosystem or an economic engine. It is a planetary thermostat component, one of countless interlocking pieces that have maintained habitable conditions for human civilization. Disrupt its deep carbon balance, and the consequences cascade upward through the atmosphere and outward across the globe. Stabilize it through decisive climate action, and we preserve not only its visible wonders, but its invisible role in keeping Earth's fever in check. We have traveled from the cosmic view of a turquoise ribbon visible from space, through bustling underwater cities, moonlit spawning clouds, hidden blue holes, ancient growth rings, glowing proteins, fragile symbioses, predatory outbreaks, bleaching silence, dissolving skeletons, accelerating heat, economic lifelines, and now to this deepest, most global secret. The reef has revealed itself layer by layer, a marvel of persistence, interconnection, and quiet power, yet one that stands at a crossroads defined by choices we make today. Now you understand why every living branch, every buried grain of carbon, every surviving colony matters on a scale far larger than one ecosystem. The Great Barrier Reef is telling us, through science, through its own changing condition, that we still have time to choose protection over loss. But that window narrows with every passing warm season. Drop in the comments below, which of these 13 secrets shifted the way you see the reef the most? Maybe it was the moonlit spawn, the hidden glow, or this final carbon story that hit hardest. Share your thoughts. Your voice adds to the conversation that helps keep places like this alive. Protect our oceans, cherish the reef, and keep learning. See you on the next dive!